So in this part of our notes, we're going to take a look at population dynamics, and we're going to take a look at first distribution patterns. We know that organisms can be arranged in clumps, they can be uniformly or evenly spaced, or they can be spaced at random. So if we have organisms in clumps, that usually means they're traveling as herds or they're in schools. If we have organisms uniformly spaced, it usually has to do with they've established a territory. And then at random, we can see organisms like dandelions where they just disperse seeds, and wherever they land is where they're going to germinate. This next question that you're going to see is one that was released from the 2014 test. So go ahead and take a second to pause the video and read through that, and we'll determine what the correct answer is. If we take a look at the answers here, and we're trying to figure out which species has the most even distribution of nests, if we just determine the area of the forest, that's not really going to tell us about the distribution of the nest. If we take the total number of observations collected during the studying period, again, that's really not going to give us an indication of nest distribution. So when we take a look at C, and it talks about standard deviation of the nearest neighbor distances for each species. If you think about it, we can calculate a mean distance, and then when we calculate standard deviation, it's basically going to give us an indication of how much variation there is between nest distance. C is your answer for that question. So next we're going to take a look at survivorship curves. And survivorship curves just basically shows us the mortality rates of organisms throughout the different stages of their life. So we've got type 1, type 2, and type 3 survivorship curves. So let's take a look at the diagram and see if we can understand the diagram, and then we'll fill in our notes here. Individuals that have a type 1 survivorship curve, what this is basically saying is throughout their life, so at different ages, there's different mortality rates. If you notice, we have really high survivorship from infancy, and we have a high increase in mortality rate whenever we reach the end of our lifespan. That's the shape that you're going to see on a type 1 survivorship curve. So high survivorship for the majority of the life, but rapid decline late in life. Organisms that have a type 2 survivorship curve, you're going to see a constant mortality rate over time. Organisms are just as likely to die when they're young versus whenever they're older. And then you have the type 3 survivorship curves. What happens in that situation is organisms that are relatively young have a high mortality rate. They do not survive well whenever they're young. But if organisms can actually reach an adult stage, then they have high survivorship. Now that's kind of tied into our life history patterns, which could also be called our life strategies. There's two types of life strategies. There's the R and the K. And sometimes we call them R-selected individuals or K-selected individuals. If we think about those organisms that show this type 3 survivorship curve, those species are usually laying a high amount of eggs, or they have many offspring. There's high growth rates. They reproduce early in life. There's little to no parenting, and they have really short lifespans. Organisms that are selected usually have a type 3 survivorship curve. When we look at individuals that are K-selected, like humans, we have few young, there's high parental investment, we have long lifespans. And populations that are K-selected usually fluctuate around carrying capacity for that environment. Our K-selected individuals usually show a type 1 survivorship curve. To kind of help you remember the survivorship curves and the life strategies, I like to think of we're number 1, because we have the type 1 survivorship curve, and we're special K. Just an easy trick to help you remember. Next, we're going to take a look at population growth curves and the stages of population growth. Now, before we do that, I want to make sure you understand what the slope of this line means and how it relates to birth rates and death rates. When we have a population growth curve, anytime we see the population increasing, then that means that we're having a higher rate of birth, so higher birth rate compared to death rate. So we have a higher birth rate, we have a lower death rate. So that's why the population is increasing. When we have zero population growth, so that means the birth rate is equal to the death rate. So if there's 4 organisms born, 4 dies, you're going to have zero growth rate. Anytime we have a decrease in population, that means that there's a higher death rate than there is a birth rate. Let's look at the different stages of population growth. Whenever you have a population that's becoming established, then there's going to be an increase in population, but it has a very low rate of increase. So this is what we mean by initial growth. There is growth, but it's low growth rate. And it's simply due to there's not a lot of individuals that are able to reproduce. So if you start off with 4 rabbits, the population is going to go up, but you only have a couple rabbits that are actually reproducing. You're going to have that low population growth. But in this situation, we do have a higher birth rate than death rate, so that's why the population is increasing. You can see how the slope of the line changes. We're getting a higher growth rate. During this phase, this is called exponential growth. So there's a high growth rate simply because the population has become established and there's plenty of organisms to reproduce. So in this situation, our birth rate is exceeding death rate, so population is going up, and there's plenty of resources. There's not a lot of limiting factors acting on the population. When the population nears carrying capacity, and this line represents carrying capacity. Carrying capacity is usually abbreviated with a K. Carrying capacity refers to the maximum number of organisms that can be supported by the environment at that time. Just for this example, let's say the carrying capacity is 80 for this population. So when this population is nearing carrying capacity, that is whenever times are getting tough. Because of the high population, there's going to be a lack of food. There's a lack of habitat. There's going to be increase in disease. There's going to be an increase in predation. And so that's why our growth rate is starting to decrease. When a population reaches carrying capacity, usually the population kind of fluctuates around that number. Anytime the population is above carrying capacity, like in this situation right here, population declined, so that means that there was actually a higher death rate at that time compared to birth rate. And anytime you have a birth rate that is lower than the death rate, you're going to see a population decrease. So this is what is meant by the different stages of population growth. So while we're talking about population growth and we're showing it on a graph, this is a good time to review calculating rate. Remember, rate is the change in something or the change in time, or it's the change in y over the change in x. In this situation, we're looking at a change in population over a change in time. Trying to get your eyes trained where when you see time on that x-axis, then you're probably going to have to calculate rate on that graph. If we want to calculate the initial growth rate, so let's take a look at this growth rate from 0 hours to 4 hours. And this is talking about the growth of a population of yeast in a beaker. In this situation, there's a change of 4 hours. So there's your bottom number. And during that time, it looks like the population increased about 100. So 100 organisms. So we have a population growth rate of about 25 organisms per hour. Let's look at the exponential growth rate. We can see the slope of the line change. So there's a different rate during this phase. Let's look at the growth rate between hours 4 and hours 8. Change in time would just be 4 hours. Now this is where students usually mess up. We do not start at 0 for the population. The population was at about 100 at 4 hours. And at 8 hours, let's say the population is at about 475. That change would be about 375 organisms during that 4 hours. That's what we plug into the calculator and calculate. Make sure that whenever you look at a graph showing a population change over time, that you're able to, for the different phases, discuss how the birth rate is compared to the death rate, what the slope of the line indicates. The slope of the line indicates rate. And then make sure that you can discuss some of the different factors, such as During exponential growth, there's plenty of food, there's plenty of space, there's plenty of resources, but whenever you near carrying capacity, that's whenever you see some of those limiting factors come in effect. Now, I wanted to show you this graph because this was a graph that was shown on one of your last tests. And I had a lot of students indicate that at month 6, there was a higher population than at the beginning of this experiment. This just shows you the need to make sure you pay attention to your axis. This is over time, but we're talking about the number of spots that were on a guppy. This just simply indicates at the beginning of the experiment, there was an average of 10 spots on male guppies. You have no idea how many organisms they were actually analyzing to determine the number of spots that were on that guppy. So this graph gives us no indication of population size. Sometimes when students see graphs and they're talking about organisms, they immediately think they're talking about the number of organisms, but you need to pay attention to the labels on the axis. We've already talked a little bit about limiting factors because we talked about that as populations reach carrying capacity, they're going to run out of food, they're going to run out of space, there's going to be more competition. So that's what we mean by limiting factors. This list of limiting factors can be classified 2 different ways. We can classify them as living or non-living. Remember, living means biotic, non-living is abiotic. You need to be able to look at the limiting factors and determine whether they're biotic limiting factors or abiotic limiting factors. I already have them listed here for you just to save time. Things like temperature, the amount of rainfall, natural disasters, habitat size, pollution, those things are considered abiotic. Those are non-living limiting factors. Whereas disease, which is caused by fungi and bacteria, those are considered living things, so they're biotic factors. Competition is talking about other organisms, so again, living factors. Predation and food supply, food's considered living. What you need to be able to do is predict what's going to happen to carrying capacity when different limiting factors are altered. Let's talk about things that would actually increase the carrying capacity of a population over time. In regards to temperature, whenever temperature goes up, then photosynthetic rate goes up, so there's more biomass, there's more food for that ecosystem. In general, when temperature increases, we're going to have an increase in carrying capacity. Whenever there's an increase in rainfall, again, higher rate of photosynthesis, so that would increase carrying capacity. And again, in this column, we're just looking at abiotic factors. Some abiotic factors that would decrease carrying capacity if we have an increase in natural disasters, if we have a decrease in habitat size, if there's an increase in pollution, then that would lower the carrying capacity for a population. So with the biotic limiting factors, if we have a decrease in disease, then that should raise carrying capacity. If we have a decrease in competition, that should raise carrying capacity for another population. To decrease carrying capacity, if there was an increase in predation, we'd see that. If there is a decrease in food supply, we would have a decrease in carrying capacity. Next, we can categorize these limiting factors as whether they're density dependent or they're density independent. Here's what you need to consider when you're trying to determine whether a limiting factor is density dependent or density independent. As the population changes, then the limiting factor changes as well. Then it's considered density dependent. The severity of the limiting factor is actually dependent on the population size. So as population goes up, then the amount of food available is going to change. It's going to decrease. So since it's changing, again, it's considered a density dependent limiting factor. Same with these other ones that are listed here. As the population goes up, competition is going to change. As the population goes up, the amount of disease is going to change. If the population goes up, it's going to attract more predators, or the predator population is going to go up just simply because it's acquiring more food. So all those things change. Now, just because I've listed biotic limiting factors here doesn't mean that biotic factors are always considered density dependent, and we just don't have abiotic factors listed as density independent. A good example is pollution. As the population goes up, the levels of pollution also go up. That's actually an example of an abiotic limiting factor that is considered density dependent. So let's look at some density independent limiting factors. Even if the population goes up, that's not going to change things like the amount of sunlight that reaches an ecosystem. As the population goes down, That's not going to alter the temperature of the environment. Temperature would be considered density independent. The amount of rainfall, and any natural disasters. As the population increases, then we're not going to get a greater chance of a forest fire or a landslide. So those are considered density independent limiting factors. We've already talked about population growth curves, but in general there's two types of population growth curves. One's called an exponential growth curve, and then we have the logistic growth curve. Let's start with the logistic, because logistic is the logical population growth that you're going to see in most species. Most species are going to hit a carrying capacity. So that's why I consider this logical, or this is a logistic growth curve. This can also be called an S-shaped growth curve just because of its shape. The only population that has not reached a carrying capacity is the human species. This growth curve is called an exponential growth curve because we haven't reached a carrying capacity. It can also be called a J-shaped growth curve. Now, when we look at exponential growth curves, then what we're actually looking at is what's called biotic potential. The biotic potential of a population is just the maximum rate that the species can increase when there's no limiting factor. This is the rate of increase when there's plenty of food, there's plenty of space, there's no predation, and there's no disease. So since we're talking about population growth, we're going to take a look at what's called these age structure diagrams. And so this is actually showing you the distribution of individuals of different ages in different countries, and whether those countries are going to experience rapid growth, slow growth, no growth, or even a decline in growth. In this situation, when you have a really high number at those different ages where reproduction can occur, then this is a situation where a country is going to show rapid growth. Countries that have less than those initial countries I just talked about showing rapid growth, they're going to have slow growth. But we still have the major proportion of individuals is in those younger years where they can still reproduce. Here's a population or populations that are going to show no growth over time, because basically the proportion of the population that's in the reproductive years is equal to the proportion of the population that is reaching the end of their lives. You can have age structure diagrams that will show a population decline. If you have a greater proportion of the population nearing the end of its lifespan, then you'll see a population decline. So for these age structure diagrams, you should just be able to look at them and indicate whether a population is going to increase or decrease just by the distribution of age groups. So if you look at the top of your formula sheet, you're going to see this whole area is dedicated to population growth. So there's actually different ways that we can calculate population growth, but one thing I want you to note about each of these different formulas here is that every single one of them is a way to calculate rate, population growth rate. So remember, rate is the change in something or the change in time. And we show that on a graph. We show change in something over the change in time. The change in the y-axis over the change in time. That's what those symbols mean. We can also calculate population growth rate by simply subtracting deaths from births. It gives us an indication of what the population is going to be increasing over time. If a population is showing exponential growth, so that's where there's no carrying capacity, then we're going to use this equation to calculate population growth rate. So again, it's just showing you we're calculating change in number over the change in time. And then we can also calculate population growth rate, again, change in number over the change in time. But this is a situation where a population has a carrying capacity. Remember, K means carrying capacity. So one thing that I've emphasized before is you need to be able to calculate rate from a graph or a table. Question 8, it says calculate the population growth rate between 1960 and 2000. We have a change in time of 1, 2, 3, there's 4 years there. So there's our bottom number, it's 4 years. And in that time, the population changed from 3 billion to 6 billion. So that's a change in 3 billion. When you plug that into your calculator, you can see that there's a growth rate of 75 million individuals per year. Here we have a table that's showing change in number over the change in time. So let's calculate population growth rate between 2010 and 2013. In that time, we've had a change of 3 years. So there's your change in time on the bottom. And then the change in population is 40 individuals. So that's going to give you an increase of 13.3 individuals per year. So you're just using that first or that second equation on your formula sheet. We're just looking at change in number over the change in time, and that means rate. So our rate of increase is 13.3 individuals per year. That third equation is just showing you that you can calculate rate, again, change in number over the change in time, by determining births minus deaths. Example C, it says you have 350 falcons in a forest. During that time, 50 falcons are born and 30 falcons die. Determine the population growth rate. So change in number over the change in time equals 50 births minus 30 deaths. So that population is increasing by 20 individuals each year. Next, we're going to get into calculating growth rate for a population that's showing exponential growth. Whenever you start to see words like maximum per capita growth rate, or if you see a number that has a decimal, then you need to know that you're probably going to be using this equation right here. As long as that problem says nothing about a carrying capacity, or it says that the population is experiencing exponential growth. Here we have a population that has initial population size of 1,000. Now, over here, if you haven't noticed, there's the symbols that tell you what these different symbols mean. N just refers to population size. So we're starting out with a population of 1,000. And it says there's a maximum per capita growth rate of 0.6. And that's defined for you right here. That's telling you that this number is 0.6. So we're just going to take 0.6, which is our max, times 1,000, and that's going to give us our population growth rate. We're getting a change of 600 individuals per year. Our max is going to be given to you in a problem, or it's even something that you can calculate if they give you per capita birth rate and they give you per capita death rate. So in that situation, all you have to do is take that per capita birth rate, you subtract that per capita death rate, and that's going to give you your Rmax, or your maximum per capita growth rate of a population. Now, we talked about on that population growth curve that rate changes over time, and it has a lot to do with population size. We have a low rate of growth when a population is experiencing that initial growth because there's not a lot of organisms reproducing. And as you get more organisms to reproduce, then that population growth rate goes up. To kind of give you an example of that, we're going to look at two populations that are going to start off with different numbers of individuals. So one's going to start off with 12 individuals, the other one's going to start off with 160 individuals, but they have the exact same per capita growth rate, or the same Rmax. In this situation, we're plugging in 0.4 times that initial population, which is 12, and I'm just using this formula, that fourth one, which is population growth rate is equal to Rmax times the initial number of the population. The population is going to add 4.8 individuals per year, or per time, depending on what that time is. It's not always necessarily year. In this other situation, we have the same Rmax, but we're starting off with 160 individuals. So just because there's a higher number of organisms that can reproduce, you're going to have a higher growth rate. There's 64 individuals that are being added to the population per year. Sometimes you're going to be asked to calculate what's the growth rate, what's the change in N over the change in time. Sometimes a problem is going to ask you what is going to be the population size for the next generation, or at the beginning of the next year, what's going to be the new population size. Going off of what we we're just calculating up there with D. We're going to start off with a population that starts with 1,000 individuals, and they have a per capita growth rate of 0.6. So we are determined in problem D that that's going to cause an increase of 600 individuals per year. These individuals are added to the population, and it started off at 1,000. That's why I have 1,600 here, is because I simply added those new individuals to that initial population size. So in generation 2, we're starting off with 1,600 individuals, not 1,000. We still have that same per capita growth rate. So when we plug that into that exponential growth equation, we're going to see that in this situation, our growth rate, which is again change in number over the change in time, is equal to R max, which is 0.6, times 1,600. We're going to add 960 individuals during that time. To figure out the initial size of generation 3, then we're going to have to add 960 to that initial population size. So in generation 3, we're starting out with 2,560 individuals. So you can see how growth rate is actually affected by the initial population size. Now we're going to solve for R max. So we're going to manipulate that equation a little bit. It says a population of 80, so there's our initial population size. So there's N. It says it has a growth rate of 60 individuals per year. So what they're actually giving you is they're giving you a change in number over the change in time. So that's the growth rate. It says what is the maximum per capita growth rate? So you're basically solving for R max. In this situation, we're going to plug in our numbers into that exponential growth equation. Our change in numbers is 60. We're trying to solve for R max. I'm just going to put an R there. And they gave us N, which is 80. Then to solve for R, we just have to divide both sides by 80. 60 divided by 80 gives us 0.75. So that's the R max value. Here's a situation where they're giving you R and it's a negative number. That means that the population is going to decrease. So we're starting out with 190 gray tree frogs. There's N, and there's your R max right here. So you just plug it in to see what's going to be the change in population during this time. So we have a negative 0.093 is the R max. Times it by that initial population size. Starts off with 190 frogs. In this situation, we're getting a change of negative 17.6. Let's go ahead and round it to, we're dropping by 18 individuals. If you put 18 individuals down as your answer, you're going to get that answer wrong, because it doesn't want to know what is the population rate of decrease. They're wanting to know what's going to be the population size for that next year. If we started off with 190, we're subtracting 18. 172 is your answer, not 18. Remember those two different population growth curves I showed you? There's exponential growth and there's the logistic growth curves. Whenever a population is experiencing logistic growth, that means that there's a carrying capacity in that ecosystem. Remember, that's represented by K. Anytime a problem is talking about there's a carrying capacity, then you need to know that you're going to use this logistic growth equation. It says in a mouse population of 1,000, so they're giving you N. There's the initial population size. It says the maximum per capita growth rate is 0.6. There's your R max. And it says the carrying capacity of the population is 8,000, and that's K. And again, on this side of your formula sheet, it's telling you what all those different symbols mean. So let's plug that into the equation to see what the growth rate is going to be. Again, we're trying to calculate growth rate. Change in number over the change in time. Our max is 0.6. The initial population started out at 1,000. And then in this part of the equation, we've got carrying capacity minus the initial population size. So it says carrying capacity is 8,000. And there's 1,000 in that initial population size. And we're going to divide by the carrying capacity, which is 8,000. So again, I'm just plugging those numbers into the equation. Whenever you simplify, this area equals 0.875. We have 0.6 times 1,000 times 0.875. And that tells us that this population when there's a carrying capacity, is going to have a growth rate of 525 individuals per year. Again, for these problems, they might ask you what's the growth rate, or they might ask you what is the population size at the end of the year or for the next generation. Again, we just calculated, I'm using these exact same numbers, and it says there's a carrying capacity of 8,000. We've already pretty much done the first part of this table, and we calculated there's 525 individuals being added per year. To start off generation 2 or year 2, then we've got to start off with that new population size, which is 1,525. So then we're going to plug that into the logistic growth equation. We have 0.6 is the Rmax, and our initial population size is 15,025. Those numbers moving are messing with me. And again, we have the same carrying capacity, which is 8,000. Initial population size is 1,525. And that's divided by the carrying capacity. When you plug all that into the calculator, then that shows you that the population is going to go up by 741 individuals. Again, we're going to have to take this number, we're going to add it to what we started off with at the beginning of population 2, and that gives us a starting population of 2,266. So on the 2013 test, they put this problem on there. So go ahead and take a second to pause the video and read through that. Here's a situation where you're given a grid-in problem and there's no formula on the formula sheet associated with this. When that's the case, you're just going to use a simple mathematical process to solve this problem. When I started looking at this equation, or looking at the data that was given in this chart, I kind of started sorting in my head, even without really reading what they said here, is that there was 14 individuals to 88. There's 180 individuals to how many? So whenever I write the data like this, you probably can see that you're just going to do some cross multiplication. So in this situation, we're trying to solve for x, or what was that population size. So we have 14x is equal to 180 times 88, and that equals to 15,840. To solve for x, you're going to divide by 14 on both sides. X equals 1,131. Okay, but let's take a look at this problem and kind of relate that to some of the stuff that we wrote down here. It says the mark recapture method assumes that the proportion of marked individuals in the recapture group. That is this. There's that recapture group. We got 14 were marked out of 88 that were recaptured, is equal to the proportion of marked individuals in the entire population. So that's talking about this side. So basically, here's what happens. To estimate population size, scientists use what's called this mark recapture method. What's kind of confusing about this data table is this is actually what scientists figure out first. They capture as many individuals as they can, and they mark all of them. So in this situation, they caught 180 organisms, and they marked them all, and then they released them. And so then they come in months later, years later, and they capture, again, as many of this population as they can. They caught 88 individuals, but only 14 were marked. Then you can just simply set up where you're showing that the proportion of the marked individuals in the recapture group is equal to that proportion of marked individuals in the entire population, and you're basically determining how many of those organisms are in that population. So that's the mark and recapture methods. Let's do another problem. It says initially 30 sharks were caught and tagged. At a later time, 60 sharks were caught and 10 were marked. Estimate that population size. Again, we caught 30, but we don't know how many total is in that population. We just caught 30 and we tagged them. So then later on, we come back and we catch more sharks, and we end up catching 60, but only 10 of the 60 were marked. So again, we're going to cross multiply here. So 10x equals 1,800. To solve for x, divide by 10 on both sides. X equals, there was 180 sharks in that population. Next, we're just going to get into how human activities can affect either positively or negatively the diversity on the planet. So we've already talked that the number one cause of extinction is habitat destruction. And remember, whenever habitats are destroyed, organisms can't just move to a new habitat, or they cannot just switch to a new food source. And also, we talked about in the evolution unit that if there is a drastic change to the environment, that's going to lead to population decline, or it's going to lead to death. It can possibly lead to extinction of a population. In order for evolution to occur, those changes in the environment have to be very gradual, because evolution takes time. So here's some activities that negatively influence the biodiversity or affect the extinction rates on our planet. Habitat destruction, just simply due to development. And what we mean by that is simply clearing off ecosystems for housing, industry, for fields to grow crops. There's habitat destruction due to harvesting of natural resources. When we remove trees for building materials and other supplies, whenever we are trying to mine natural resources, we can destroy habitats in that process. Humans also destroy habitats by the introduction of exotics or introduced species. So sometimes it happens on purpose, sometimes it happens on accident, but we have a tendency to introduce exotic species, which can cause ecosystems to collapse. And we have habitat destruction due to pollution. And we'll talk more about the different types of pollution that you need to know for the test. But relating that to that ecological pyramid, if we go into an ecosystem and we bulldoze and remove the majority of the plant life, then you can see we're going to set up a situation that looks, for a period of time, it's going to look like this. We're going to have very little plant biomass. We're going to have a lot of those upper level consumers. This is very unstable. And you know, there's going to be lots of starvation and death. And ultimately, you're going to get back to an ecological pyramid that is wider on the bottom. But since you've restricted that habitat size, you're going to have very few producers, even less of those upper level carnivores. And it just simply leads to a very unstable ecosystem. Because in this situation, there's just not a lot of biodiversity. And less biodiversity, the more unstable an ecosystem. So now let's look at the different types of pollution. We're going to focus on acid rain, global warming, the hole in the ozone layer, biological magnification, and eutrophication. We've already talked about eutrophication. With acid rain and global warming, both of those are due to the burning of fossil fuels. Whenever we burn coal or use gasoline in our cars, what happens is there are chemicals that are going to be released up into the atmosphere. There's sulfur oxides and there's nitric oxides that are released whenever we burn coal. Those molecules go up into the atmosphere, and then whenever water condenses, whenever it gets cooler, then these chemicals are water soluble, and so they come down in the rain. We've already talked about organisms have to maintain a homeostasis. All organisms have a limited pH range that they're tolerant to. So in this situation, terrestrial means land. What happens in this situation, whenever that rain comes down with that lower pH, then the soil becomes acidic and the plants can't survive. Again, you're eliminating that biomass on that first trophic level, which is going to affect all the other organisms on the next trophic levels. With the freshwater ecosystems, all this water eventually runs into a freshwater ecosystem, and so that water becomes too acidic, and so you're going to have the loss of plants, and fish can't tolerate those lower pHs. Now, when we're talking about global warming, we're talking about there's a layer of gases that kind of blanket the Earth, and those are known as those greenhouse gases. So some examples of greenhouse gases. CO2 is a greenhouse gas, and methane is a greenhouse gas. Even water vapor is considered a greenhouse gas. Now, to be a greenhouse gas, what that means is that you're a gas that will keep heat in that Earth's atmosphere. Whenever we burn fossil fuels, we burn organic molecules, it releases carbon as CO2 up into the atmosphere. We basically increase this layer of greenhouse gases, and we're increasing the amount of heat that is trapped close to the surface of the Earth. Now, I want to make sure you understand that the greenhouse effect isn't a bad thing, because we do rely on these gases to keep our atmosphere at a comfortable temperature. Some heat being trapped is good. So think of a thin blanket on a warm night. That's a good thing, but you don't want your blanket too thick, because then it's going to trap too much heat and you're going to heat up too much whenever you're sleeping. The greenhouse effect isn't a bad thing. It's just we need to make sure that we don't have too many greenhouse gases trapping too much heat up in the atmosphere, leading to global warming, which is the increase in global temperatures over time. Now, the hole in the ozone doesn't really have anything to do with burning fossil fuels. It's due to a man-made chemical. We talk about chlorofluorocarbons. Chlorofluorocarbons were used as solvents, they're refrigerants, propellants. For example, what we mean is, think of your aerosol cans. So whenever you would spray like hairspray in an aerosol can, chlorofluorocarbons were put in that can, and they're propellant. They help that hairspray come out. Well, when you spray hairspray in the atmosphere, you're also releasing these CFCs that are in those aerosol cans. So what happens is these CFCs are traveling up to the ozone layer. Let's talk about what that ozone layer is. Ozone is actually just 3 oxygen atoms stuck together. And what's kind of interesting about ozone is if you have ozone right next to the surface of the Earth, that's actually a major component of smog. And so it can cause respiratory distress and asthma when you breathe in ozone. So it's actually a pollutant whenever it's close to the Earth's surface, but when it's up here in the atmosphere, it acts as a filter for the sun's UV rays. It doesn't block all the UV rays, but it blocks a proportion of them. These CFCs are floating up into the ozone layer, and they were reacting with the O3, and they were breaking it up. And so that was allowing more of the UV rays to strike the surface of the Earth. What that leads to is a little bit of global warming, because we're actually having more energy make it to the surface of the Earth. But our biggest concern is that the increased exposure to UV rays increases mutation rates. So thinking about skin cancer. We've already talked about eutrophication, and again, we were talking about it when we were talking about the nitrogen cycle and the phosphorus cycle, because these are elements that lead to increased plant growth. Whenever we put down fertilizers on our yards, whenever we have large-scale animal operations, so think of chicken farms and pig farms, that waste, when it rains, runs off into freshwater ecosystems. And so it causes algae, which is a plant, to increase in population. What eventually happens is that algae is going to reach a carrying capacity, and it's going to die and decay. But oftentimes what can happen is we get these algae blooms, which is just a high population growth of algae, and it blocks the sun. And so if there's any plants that are down here, then they die as well. So what happens in that situation is the decomposers are going to consume that organic molecule, and they're going to turn it into ATP. So they're going to perform respiration, and that uses oxygen. So we're showing oxygen being used by those decomposers. And so that's what leads to these fish kills, just simply the depletion of oxygen. But it all starts off with those excessive nutrients running into those freshwater ecosystems. Now, in biological magnification, what is occurring is that we have chemicals like pesticides, heavy metals that are introduced in the food chain. So what happens is these chemicals are put down or sprayed for whatever reasons, and they end up in water supplies, or they end up on the plants, and then our consumers take in these chemicals. So they eat the plants that have these chemicals on them. Then we end up with these chemicals becoming concentrated in the tissues of these organisms. Now, as your upper level carnivores eat these organisms, what happens is you end up with a higher concentration of these chemicals in these upper level organisms. This higher concentration in their tissues. And when the concentration reaches certain levels, it can actually cause disruptions to homeostasis. It can affect different physiological processes that are taking place in those organisms. This was one reason why the eagle population almost became extinct, is because we were spraying an insecticide, which is called DDT. As these eagles were consuming organisms that contained this DDT in their tissues, it became concentrated in those eagles. And whenever they laid their eggs, those eggshells would be paper thin and they would break upon laying them. The eagle population wasn't able to reproduce because of this exposure to DDT. And like I said, it almost caused them to go extinct. Since we're looking at changes to ecosystems, this is a good time to talk about primary and secondary succession. There's two main types of succession. If succession is occurring, basically we're just seeing a change in species structure over time. Or so it's kind of like the development of land and the species that are going to be on that land over a period of time. When we're talking about primary succession, then you basically need to think of an area that is brand new, newly established, and that usually takes place when we have volcanic activity. All this rock is placed down, but there's no soil there. The big difference between primary and secondary succession is that there's not going to be any soil in this location. So what happens over time is you get these pioneer species that start to grow on that rock. And those are the lichens and the mosses. Weathering over time, the breaking up the rock, you get fine particles, and eventually get the development of soil. So after we get these pioneer species, then over an extended period of time, then there's a little bit of soil development, and it's enough to support some of your grasses. And then again, later on in time, you're going to get some bigger shrubs, and then eventually to trees, and then eventually to really tall trees. And this is what is noted as the climax community. So this is the final established stable community that's going to develop over time due to succession. This is just representing over time. So it takes many years for an area to establish that climax community. In secondary succession, think of a habitat that has soil and then something happens to it. So it can be destroyed by fire, it can be cleared off for farmland. But the big difference is there's soil there. We're not starting off with rock like we see after a volcano explodes. So there's soil there. So pretty quickly you're going to see plants are going to start to develop, your grasses, and eventually it leads to your shrubs, and again to your bigger trees and to that climax community. The difference is no soil with primary succession, but there is soil initially. We see areas going through secondary succession. So we've been talking about negative influences by humans, but we do have the ability to positively influence the environment. Here's some of these different techniques that we do to try to preserve biodiversity. One thing we can do is we can recycle. We have habitat that's preserved, so habitat preservation. The government sets up parks and recreational areas that cannot be developed, and they're trying to maintain natural habitats and maintain biodiversity. We can restore habitats, so habitat restoration. We try to educate people so they can learn how to minimize our impact. We limit or we ban certain chemicals. So for example, DDT is now banned in our country, but it's not banned in other countries. So it is finding its way into food chains. We will tax companies that are releasing too much pollution, or we fine them, or we even lower their taxes if they're a green company. We restrict hunting. We have endangered lists. Those organisms are not allowed to be hunted at all. A lot of our bans and regulations are put in place to protect what's called keystone species. So here's the important idea to understand about keystone species. The effects of the keystone species on the ecosystem are disproportionate to their abundance. What that means is when you look at the amount of biomass or the number of those organisms, it's actually really low. But these organisms, despite having really low biomass and low numbers in ecosystems, they're highly important. We believe keystone species, if they go extinct, would actually lead to the collapse of entire ecosystems. One example of a keystone species is salmon. Now, because of their niche, because they interact with so many different organisms, we know that if salmon go extinct, then that entire ecosystems are going to collapse. So again, salmon is an upper level predator. There's really a low amount of biomass in salmon compared to their importance. This graph just kind of indicates that as long as these keystone species are present, then we're able to maintain biodiversity. Species aren't going to go extinct. As a matter of fact, this is showing an increase in the number of species in an area as long as that keystone predator is present. But here's a situation where the keystone predator is extinct or is absent, and you can see biodiversity, you can see this ecosystem collapse as the number of organisms decrease because so many of them are reliant on that species. They're influencing the community and it's disproportionate to their biomass. Something to probably have in mind is a good example of what different organisms give or supply to us. When we talk about organisms are important for biodiversity, what do they supply for us? Think about the food that is provided by our natural environments. Think of how many people are employed by agricultural jobs related to food production. Insects have an extreme agricultural value. If something was to happen to bees, then we would see a loss in about $10 billion worth of crops. They're important for pollination, so plants will reproduce so we can have those seeds, we can have those grains. We get lots of manufacturing supplies, building supplies. We get textiles from living organisms, cotton, sheep, building supplies, and like I said, even goods that we use in manufactured goods, like rubber comes from a rubber tree. And then we also believe that there's about 300 drugs that are still yet to be discovered. So we're talking about molecules that are found naturally and are made naturally by organisms, and we can use them to cure different diseases. Here's some examples. This rosy periwinkle, there's a chemical in it that we isolated and we treat leukemia and Hodgkin's disease. And so you can see before this was discovered, there's a 10% survival rate in this disease, and now we're up to a 90% survival rate. Penicillin comes from a fungus, and we use it to kill bacteria. The horseshoe crab, these are actually collected, and they're bled. They're not killed, they're released, but that blood contains a chemical that actually inhibits bacterial growth. We can use that to keep different medical devices free from bacterial growth. And then we use organisms for research. Some organisms, there's different bacteria that normally lives on them, and that bacteria can be harmful to us. So we can actually study these organisms and what is it about their chemical composition, or what molecules are they making that's protecting from these different types of bacteria, and maybe that's something that we can utilize in medicine.